Some viruses are going to have an envelope. Not all viruses do. If a virus has an envelope, it's called an enveloped virus. If a virus does not have an envelope, then it is termed a naked virus. They're going to have a central core. Within that central core is going to be the nucleic acid molecule, whether it's DNA or RNA, it's never going to be both. Then there's a special term given to the capsid and the nucleic acid molecule, that's called a nucleocapsid. There's matrix proteins and enzymes. They're not going to be found in all, but sometimes viruses do carry special types of proteins or things that they need. Ultimately, a virus will carry whatever it has to have that it cannot manipulate from its host cell to use. Classification. We've talked about genetic material. We've talked about what it infects. We've talked a little bit about size. Now we're going to talk about shape. You definitely need to know all of these. What is a viron? It's basically just a virus that's outside of the host cell. There's a capsid. That's going to be from here. That's one of the coverings. The capsid is a protein coat. What does it do is it protects the nucleic acid. Sometimes there's an envelope. That envelope is going to surround the capsid. When something has an envelope, it's usually considered to be a lesser resistant virus. That envelope is typically composed of a phospholipid bilayer. Not all viruses have an envelope. If it doesn't, they're going to be termed a naked virus. Where does the envelope come from? It comes from the cell it's infected. Viruses have spikes. Those are going to be proteins that are coming off of the edges. Those protein spikes will interact with the receptor protein, and that's basically going to dictate hosts, what type of cells the viruses can infect. This is showing you a schematic of a capsid. Nucleic acid is inside, capsid is surrounding the nucleic acid. Altogether, this is called a nucleocapsid. And we have 3 different capsid structures, and you're going to need to know all of them. Capsid morphology, again, they provide protection for the viral nucleic acid. It is also a means of attachment to host cells when that capsid degrades. It's usually when the virus is inside the cell, releasing the genetic material. It's composed of a variety of different proteins. A lot of times those proteins are protein subunits, or just protein pieces, and they come together to make a capsid-mere. So a capsid you could call is like a polymer, okay? And it's composed of repeating units of known as monomers. And in this instance, that monomer is going to be a capsid-mere, okay? So capsid-meres may be made up of single or multiple types of proteins. These are the 3 capsid structures. You do need to know these. It's saying again, the capsid-mere is a subunit that's going to compose the capsid. So if you see this is the helical capsid. This individual purple thing is one capsid-mere. And you can put together a whole bunch of capsid-meres, it's going to form a capsid. Okay? We have 3 capsid structures. You have helical, polyhedral, more particularly known as icosahedron, and then you have complex. You need to be able to identify this. So the classification of capsid here is complex. Okay, even though it does have icosahedral head, and then it's going to have this sheath, which is actually going to be helical, it has both. So bacteriophage fall into a classification known as a complex virus. This is polyhedral, also known as icosahedral. This is helical. So we're going to talk about them individually. You definitely need to be able to identify them. If I give you pictures, you should tell me what's going on. So capsids, general structure of viruses. All viruses have a capsid. What is a capsid? It's going to be a protein coat that encloses and protects the nucleic acid. So inside is the nucleic acid, on the outside is the capsid. This entire structure together is going to be called a nucleocapsid. And this is a naked capsid. It does not have an envelope. So some viruses have an envelope that's going to surround it. That's going to be called an envelope virus. So if I put this picture on your exam, you should be able to label it. You should say that this is nucleic acid. You can't just say it's DNA because what if it's an RNA virus? So you've got to say it's nucleic acid. This is a capsid. The capsid is composed of capsid-meres. This is an enveloped virus. You should be able to label all of the points on here. These are called spikes. They're not called pokey things or fingers or receptor proteins. They're called spikes. And you do need to know that. So if I put this on your exam too, you should also know right off the bat that that is a virus. Okay, it's an enveloped virus. You should be able to do all of that. All right, the 3 different types of capsids. A capsid is composed of many identical protein subunits called capsid-meres. Capsid-meres come together to make capsids. We have helical capsids, icosahedral capsids, and then complex, which is a mixture of 2. Helical capsids are typically rod-shaped capsid-meres bound together that's going to form almost like a cylinder, and then the nucleic acid is protected within the middle of it, or it's kind of intertwined. Icosahedral kind of looks like a diamond. It's a 3-dimensional shape. It's got a lot of faces to it, but it's typically always represented like this. okay, with three-dimensionalness added to it. Then you have a complex, that's going to be a mixture of the two. The reason why the bacteriophage is called a complex virus is because it looks like this. You see it has the capsid head, this is the capsid head, and then this is called a sheath right here. This is a helical structure, and this is going to act as a pump, and it's going to help pump this genetic material inside of the cell. Okay, so this is called complex. Why? Because it's a mixture of the two. All right, helical. Let's talk about helical. Continuous helix of capsomeres. Again, it forms this like cylinder structure, kind of looks like a tube, and then the DNA is strung, or RNA, the genetic information, genetic material, is strung through it. It's made of protein, the individual units, so this one little blue thing is called a capsomere. Okay? So again, this is showing you, this is just this flipped over. So what's this blue thing? A capsomere. This whole thing right here is a capsid. The capsid plus the nucleic acid is going to be called a nucleocapsid. This is one of three structure classifications. Icosahedral, this is also known as polyhedral. The capsomeres are typically going to be arranged equilaterally, forming like triangular sides. It's three-dimensional. It's typically going to be 12-sided, and it's usually relatively evenly spaced. This is another representation of it. This is basically showing you the protein spikes, but this is done by a computer, so it's really not real. Complex. Virus. These are atypical viruses. So things like the pox virus fall into this category. So do bacteriophages. You need to be able to identify these. Not this guy, because that looks like it could be anything. So he's not really a fair question. But this one's more than fair, because this looks helical. This is obviously a icosahedral. So what's the classification of this? Complex. And that's capsid structure. You don't just go, the capsid is icosahedral. Well, because what about this? This is helical. So the classification of structure there is going to be complex. So, T4 bacteriophage. You need to be able to label this and tell me exactly what it is. And there's a number of different questions I can ask you about this. I can ask you to label everything. You should be able to label everything. These are called tail fibers. These are not called legs. Okay? And these are not called teeth. Okay? So that's really important. You should be able to identify all the parts. I could ask you a question about what classification of microbe is this. It's a virus. See where you're like, wait a minute, it's not a microbe because it's not alive. Viruses do still count as a microbe. So I see where you're headed with that, and I'm happy that you went there. But it is, even though it is considered to be a microbe, because it's one of the most smallest microorganisms that we know of, it is acellular, so it is not a living thing. So I see where you're going with that. But it is a virus. What is the virus's specific name? The specific name of this virus is the T4 bacteriophage. You need to know that. So it is a virus. It is a T4 bacteriophage. What cell does it infect? It infects E. coli. Then there's a number of questions I can ask you about that. I could ask you what classification of cell does this infect? That correct answer would be prokaryote. I could ask you about what organism does this infect? That's going to be E. coli and not prokaryote. Okay, so again, you've got to remember all of those terms. Okay, you got to be able to answer the question, well, answer what's being asked of you. And what is the classification of structure? Complex. It's a complex virus. It has an icosahedral capsid, and then it's going to have helical here. And this is going to be unique to bacteria, so it does not infect us. This is showing you an envelope to virus that's going to have the helical capsid on the inside. The nucleic acid is within that. And then it's got an envelope. Where was this envelope obtained from? It's typically going to be obtained when it buds off of the host cell. Functions of the capsid envelope: it protects the nucleic acid when the virus is outside of the host cell. It helps the virus bind to cell surfaces. How? Based on the spikes. The spikes can help with the attachment process and the affinity. It also helps with the penetration process. When something has an envelope, the cell welcomes it in right off the bat, because the envelope of this virus comes from a cell that it's trying to infect. Okay, not the same cell, but a virus buds off, let's do HIV, for example. It buds off of the CD4 T cell. It buds off, now it's mature, it wants to go infect a new T cell. The T cell interacts with the receptors and says, we've got the same stuff, I should let this guy in. As it pulls the HIV virus closer, it realizes that the envelopes are exactly the same. And then that's at the point of no return. HIV is then fuses with the envelope because it's the same, it's a lipid bilayer. It's the envelope of the virus is part of the lipid bilayer of the previous CD4 cell, which is going to allow it basically just to like merge right in. Then when it merges in, that envelope is gone, and what do you have? You're going to have the nucleocapsid and the nucleic acid. And then it starts the process of central dogma, okay? And that's how it's going to generate more viruses. So, the viral envelope. Mostly animal viruses are going to have a viral envelope. They're acquired when the virus leaves the host cell, phospholipid host, and then there's proteins. There's viral proteins, like Ebola has these glycoproteins that are relatively unique to it. The exposed proteins on the outside of the envelope are going to be called spikes. Spikes are proteins that interact with receptors. The receptors are on the host cell. That's important that you guys know all of that stuff. They're essential for the attachment of the virus into the host cell. The viral envelope, where did it come from? Again, it was obtained when the virus bud off the original host cell. Envelope is a portion of the membrane system of the host, that would be the cell membrane. It is composed of a phospholipid bilayer and then a collection of proteins. Some proteins are virally encoded, which means the viral nucleic acid was encoded to generate the protein spikes that are unique to this virus that is not from the individual cell. The envelope proteins and glycoproteins often plays a role in host recognition as well. So this is just showing that you have the DNA course. This is a DNA virus where you're going to have a capsid that's going to protect it, and then you're going to have an envelope. This is not the HIV virus, but it's going to have relatively the same structure. This is a representation of the bacteriophage. You should be able to identify this picture as well. If I put it on your exam, it's going to have a capsid. There's going to be nucleic acid on the inside of it. This is going to be a helical capsid right here. DNA will pass through this. You should be able to identify all the parts in there. The name of that virus is T4 bacteriophage. You need to know that. The type of organism it is, type of classification of microbe would be a virus. More properties of a viral envelope. They're typically sensitive to nonpolar solvents because they destroy the virus's membrane, thus its ability to infect. That's going to be an inactivated virus. That's what Lysol does. When Lysol says it kills the virus, this is what it really means that it does. Naked viruses, though, they are usually resistant to nonpolar solvents because they don't have that phospholipid inside of them. So it's harder to denature their proteins. How can you determine if a virus is going to be enveloped or naked without the visualization of the virus? It's quite simple. You can treat it with a nonpolar solvent and see if it is active or if it's inactive. If exposure to a nonpolar solvent renders the virus inactive, then it's most likely an enveloped virus. Okay? Types of viruses, you should be able to identify these based on the pictures. Again, it's just showing you complex virus. This picture is really not that fair. This one's really not fair. So if I was going to show you a complex virus, it would be something that looks like a bacteriophage versus enveloped viruses, icosahedral viruses, and helical viruses. So if I show you pictures of those on your exams, you need to, one, be able to tell me that it's a virus. You need to tell me the capsid structure, whether it's icosahedral, polyhedral, helical, or complex. And then label the parts, the capsid, the nucleic acid, and the nucleocapsid, maybe even the capsimeres. Complex virus structures and functions.